Hey everyone, this is our topic on the cerebrum and higher cortical functions. I am Ophelia Ibanez and I will be discussing this with you. So this is going to be our outline for this topic. We're going to start with the fundamentals of the nervous system. We're going to proceed to the coverings of the brain, and finally we'll talk about the cerebrum, which is divided into our telencephalon and diencephalon. This is our lesson objectives. Please pause the slides so you can see what you should be getting out of this session. And this is the next. Pause again. And we'll move to our topic outline. This is going to be our topic outline. As you can see, it's a bit long, so you can take pauses in between the slides just to have time to digest everything before moving forward, so that we avoid being overwhelmed with what we're learning. And finally, there we go. Alright, so when we're ready, let's move forward and start. Alright, let's start with the fundamentals of the nervous system. We're going to be first talking about the planes and the neuroanatomic terms. I have summarized it on this table here on the side. So when we say ventral or anterior, so ventral meaning that is something on the front or the belly side, so that means it's anterior. So that's what ventral means, it's anterior. When we say something is dorsal, that is on the back side, or something that's posterior. When we talk about something that is superior or cranial, that means it's going up, superior. When something is going inferior, so that's just going to be the opposite, it's going down, inferior. When something is going caudal, that means it's going down to the lowermost position or at the tail end. So something is going caudal, okay, or going to the tail end. When we're talking about something that's going rostral, it's on the forward side or at the nose end. So think about the nose of a person, so it's going to the nose end, so that means it's going rostral. Okay, when something is medial, it is close or towards the middle. Okay, so if this is the medial part, the median line, when something is median or medial, it's closer to the middle. Okay, when something is lateral, it's towards the side. So it's going away from the middle. When we say something is ipsilateral, that means it's on the same side. When we say contralateral, it's on the opposite side. And when something is bilateral, that means it's on both sides. Okay, let's now talk about the early development of the nervous system. So in the embryo, there are three main cell layers that differentiate. So we have our entoderm, which when they differentiate, they will become our GI tracts, our lungs, and our livers. The mesoderm, which will become our muscles, connective tissues, and the vascular system. And we have our ectoderm, which will become our nervous system. So essentially, the steps is sort of summarized on this picture right here. So you can just view that, but we will be talking about it briefly. So the first thing that happens is the formation of the neural tube and the neural crest. Okay, so what happens here is the ectoderm thickens and forms a neural plate. Okay, so this will develop a neural groove and will deepen so that it is bounded on either side by neural folds. So the neural folds will fuse until the grooves will become a neural tube. As the neural tube forms, the cells from the lateral margin will form a strip and will cover the neural crest. Okay. Now these cells will migrate and differentiate into other cells that will be the different parts of our bodies, and those will be used by our bodies. Now, what's happening here in this part is the formation of our let's say, our spinal cord. All right, so it's essentially forming our nervous system. Okay, so formation of the flexures will happen, and formation of vesicles. Okay, so we have a 3-vesicle stage. We have our prosencephalon, which is our forebrain. We have our mesencephalon, which is our midbrain, and our rhombencephalon, or our hindbrain. And then finally, it will become a 5-vesicle stage. So it's going to be our telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. Okay, after that, cell proliferation will happen, which is the rapid formation of cells. These are our neuroblasts and glioblasts, and formation of meninges around the neural tube, and finally the migration from the germinal zone to the intermediate zone. So at this point, cells with common properties will aggregate or go together, and sometimes when anything happens on any part of this to form our nervous system, or even during cell differentiation, well, defects may happen to a child or to a person. So, as we've seen a while ago, and as I have mentioned, failure onto any part of those processes, let's say failure of the anterior neuropore to close, an encephaly might happen. So when the neuropore doesn't close, well, the brain will not form, or there will be an incomplete skull and cortex. Now, when there is failure of the posterior neuropore to close, something like spina bifida could happen. And on the other side, when there is a failure of migration of cells, let's say there are defects with the proliferation and migration of cells, something like dyslexia could happen, which is because of abnormal cell positioning and connections, or even microencephaly could happen, or lysencephaly could happen, which is, as you can see, it's a smooth brain. We don't have our gyri, we don't have our sulcus. And that is also because of abnormal cell positioning and connections during the proliferation and migration stage. Let's now move forward to the coverings of the brain. So this is essentially how the brain layers look like. So we have the skull first, and then an epidural space after that. Finally, we have this dura mater. After that, we have a subdural space, and then we have the arachnoid matter. After the arachnoid matter, we have the arachnoid space, and then we have a pia mater, and after that would be your brain. Let's start with our epidural space. So our epidural space contains epidural venous sinuses, and then after that we have immediately our dura mater. So our dura mater, the dura mater of the brain, has an endosteal layer and a meningeal layer. So an endosteal layer, this covers the inner surface of the skull bones. And after that, we have our meningeal layer, which is the dura mater proper. So this is a dense and strong fibrous membrane covering the brain, and this is continuous through the foramen magnum with the dura mater of the spinal cord. Let's now talk about the dural folds. So the meningeal layers will form 4 septa. We have our falx cerebri, falx cerebelli, our tentorium cerebelli, and our diaphragma cellae. So these divide the cranial cavity into freely communicating spaces that lodge the subdivisions of the brain. So you can see that in the pictures right here, so you don't have to imagine. Okay, so those are the divisions. All right. Okay, so these will also restrict the displacement of the brain associated with acceleration and deceleration when the head is moved. So let's first talk about our falx cerebri. So this is something that looks like a sickle, that is a fold of dura mater that lies in the midline between the two cerebral hemispheres. Okay, so that thing right there, that is our falx cerebri. Our tentorium cerebelli is a covering of the upper surface of the cerebellum, and this supports the occipital lobes of the cerebral hemispheres. This one right here. And then we have our falx cerebelli, which projects forward between the two cerebellar hemispheres. And our diaphragma cellae, which forms a roof over the pituitary fossa and covers the pituitary gland. Moving forward, we have our subdural space. So this contains extra-axial fluid collections that run in that area. And after that, we have your arachnoid matter. Your arachnoid matter, sometimes in other areas, this projects into the venous sinuses to form arachnoid villi. And your arachnoid villi are sites where the CSF diffuses into the bloodstream. Okay, so if we go back to the picture, we have the bone, and then we have your epidural space, and then your dura mater, and then your arachnoid mater right here. Okay? Then we have our subarachnoid space that contains CSF that is produced by the choroid plexuses. And finally, we have our pia mater, which is the covering of the brain. So it covers the brain and all the gyri and descends into the deepest sulci and even down to the cranial nerves. So this forms the tela choroidea and fuses with the ependyma to form choroid plexuses, essential for, again, CSF production. So let's talk briefly about intracranial hemorrhage. So intracranial hemorrhage is an excessive brain movement or cranial trauma that may cause cranial vessels to rupture. Now, when these vessels rupture, of course, a hemorrhage can happen, and there are different types of hemorrhages. So we have an epidural hemorrhage, where you can guess it right, it happens in the epidural space. So injuries to the meningeal arteries or veins causes epidural hemorrhage, and the most commonly damaged is the anterior division of the middle meningeal artery. So if we look at CT scans, it's going to look like a lens. So it's a lens-shaped, dense collection of blood. So it looks like a lens. Alright? And then we have our subdural hemorrhage, which again happens where? In our subdural space. So this results from tearing of the superior cerebral veins, usually a blow from the front or back of the head. Now, if we look at it in a CT scan, it's going to look like a crescent. So this one is crescent shaped. And we have a subarachnoid hemorrhage, which of course happens where? In our subarachnoid space. So this results from leakage or rupture of a congenital aneurysm or from an angioma or a contusion. So it happens in our subarachnoid spaces. Okay, let's now talk about the bulk of our topic, which is the cerebrum. Okay, so this is how the cerebrum looks like. And we have our brain or our encephalon, which is subdivided into our cerebrum or our forebrain. Then we have our cerebellum and then our brainstem. But for this topic, we will be focusing on this part. So we will be talking about first our telencephalon, which consists of the cerebral cortex, subcortical white matter, the commissures, and the basal ganglia. And we have our diencephalon, which has our thalamus, hypothalamus, epithalamus, and subthalamus. The cerebrum is the largest part of the brain, situated in the anterior and middle cranial fossa of the skull. and this occupies the whole concavity of the vault of the skull. So again, it is divided into two parts. We have our diencephalon, which has our thalamus, hypothalamus, epithalamus, and subthalamus. And we have our telencephalon, which has our cerebral cortex, our subcortical white matter, and basal ganglia or nuclei. So let's briefly talk about the diencephalon first and discuss it generally. So the diencephalon forms the central core of our cerebrum. So this is our diencephalon right here. So it is at the core. All right, so we have four parts here. We have first our thalamus, which is the central hub, and this relays and integrates the motor and sensory impulses. Next, we have our hypothalamus, which is the center of physiological processes. And then the epithalamus, which consists of our habenular nuclei and pineal gland. This is the center for integration of olfactory, visceral, and somatic afferent pathways. And we have our pineal gland that is attached to the diencephalon, and this is attached via the pineal stalk. Now, this is an important endocrine gland that influences the activity of the pituitary gland, the islets of Langerhans of the pancreas, the parathyroids, the adrenal cortex, adrenal medulla, and the gonads. And most importantly, this is in charge of circadian rhythm of the body, and this releases melatonin. Okay, and we have our subthalamus, which is the main function of this is for movement regulation. This works alongside our basal ganglia. Now let's move forward to our telencephalon. So the telencephalon forms the cerebral hemispheres. We have our cerebral cortex, which is responsible for the higher level processes of our body or brain, and also responsible for our consciousness. We have next our subcortical white matter, which is responsible for the interconnection of cortical and subcortical areas. And we have our basal ganglia or our basal nuclei, which is primarily for motor control as well as for motor learning, executive functions and behaviors and emotions. And there are collections of masses of gray matter here, and it will have the corpus striatum, the amygdaloid nucleus, and the claustrum. So you might be confused, what's the difference between the cerebrum or the cerebral cortex? Well, the cerebrum is the largest part of the brain, so the entire thing here, that's your cerebrum right there. Now, the cerebral cortex is the outer layer of the cerebrum, if you can see the picture right here. So this is essentially your cerebral cortex. So your cerebrum is made up of both gray and white matter. Your cerebral cortex is made up of gray matter. Your cerebrum has two hemispheres. However, your cerebral cortex has four lobes, our frontal, parietal, temporal, occipital. So the cerebrum controls voluntary muscular movements, and your cerebral cortex is involved in consciousness. Let's now look at the general appearance of our cerebrum. So the cerebral hemispheres are separated by a deep midline sagittal fissure, the longitudinal cerebral fissure. So the fissure contains the falx cerebri and the anterior cerebral arteries. And we have the corpus callosum that connects the hemispheres across the midline. So we saw kanina na our brain or our cerebrum has two hemispheres, and that is connected by this, which also contains the falx cerebri, and the corpus callosum connects the two hemispheres across the midline. And then we have our gyri. As you can see, our brain is not smooth, okay? It's all wrinkly. So the gyri, the cerebral cortex, is arranged into folds to increase and maximize the surface area of the brain. And those gyri right here are separated by each other, from each other by what we call a sulci. Okay, so those deep areas are our sulcus. So the hemispheres are divided into lobes, which we talked about a while ago, which are named according to the cranial bones where they lie. So we have our frontal, we have our parietal, our temporal, and our occipital lobe. Now we're going to proceed to our telencephalon. We're going to start first with the main sulci and our fissures. So our sulci are furrows, fissures are larger, deeper sulci, and our gyri are folds. So we have our first sulci, we have our longitudinal cerebral fissure, which we talked about a while ago. It separates the right and the left hemispheres. Okay, now we have our lateral cerebral fissure right here. Okay, so this separates the frontal parietal lobe from the temporal lobe. So this is your frontal lobe, this is your frontal lobe, this is your parietal lobe, this is your temporal lobe, and that is the separation from your temporal lobe right there. Okay, next we have the fissure of Rolando. The fissure of Rolando, also known as your central sulcus. Okay, this separates the parietal and the frontal lobe. So this is the central sulcus, this is your frontal lobe, this is your parietal lobe. So the central sulcus, or the fissure of Rolando, is the separation. So this is an important landmark because anterior to that is your precentral gyrus that houses the motor cortex, and posterior to that is your postcentral gyrus, which houses your synesthetic area, which both of this, we're going to be talking about this fully in detail in the coming slides. Next we have our parieto-occipital sulcus. This separates the parietal lobe from the occipital lobe. So if this is our parietal lobe and this is our occipital lobe, okay, this area right here, the separation would be our parieto-occipital sulcus right there. And then we have our calcarine sulcus, which separates the occipital lobe into our inferior lingual gyrus. This is our lingual gyrus, and our superior cuneus. And the calcarine fissure or sulcus is the one in red right here. Okay, so there is no definite delineation from the splenius of the corpus callosum to the occipital pole, and this is actually more visible in the medial view of the cerebral hemisphere. Okay, finally we have our circular sulcus, or our circuminsular fissure, which surrounds the insula and separates it from the parietal, frontal, and the temporal lobe. So if we look further inside, this right here is our insula. So the insular cortex, aka also known as the island of Ryle, found deep within the lateral sulcus of the brain. So this is one of the least understood part of the brain because of its location, but it is suggested to have roles in sensory motor and cognitive functions. Okay, before moving forward, let's apply this clinically. So what are the fissures for? What's the significance of that? Well, our body is, or our brain has left and right hemisphere. So each hemisphere would have a main role or main function. So our left hemisphere would be for sensory stimulus from the right side of the body, motor control of the right side of the body, speech, language, and comprehension, analysis and calculations for time and sequencing, recognition of words, letters, and numbers, while our right hemisphere, sensory stimulus from the left side of the body, motor control of the left side of the body. It's for creativity, for spatial ability, for context, perception, recognition of faces, places, and objects. So it's actually a crossed pattern. All right, there is that cross pattern. Now, what happens then if, let's say, for example, a client would have a stroke on the left hemisphere? Well, then what will happen is the right side of the body will be affected. So sensory stimulus of the right side of the body will be affected, motor control of the right side of the body will be affected, speech will be affected, difficulties in analysis and calculations, in sequencing and recognition of words, letters, and numbers. So if you look at this table right here, if someone would have a left hemisphere lesion, they would have a right side hemiplegia, a right side sensory loss, and all that I have mentioned. So we have Broca's aphasia, Wernicke's aphasia, global aphasia, difficulty processing verbal cues, while a right side lesion would have a left side loss of strength, left side sensory loss, visual perceptual impairments. We would have unilateral neglect, agnosias. So I know there are words that we're not very familiar with yet, but we will try to discuss this as we move along, and hopefully we become more familiar with them. Alright, now let's move along the cerebral cortex. So the cerebral cortex, this forms a complete covering of the cerebral hemisphere, and this is the highest level of the central nervous system. So it is actually composed of gray matter and about 10 billion neurons. As you can see, the surface again has convolutions or gyri separated by sulci, as we've discussed before, and the functions in association with the lower center. So we have the three levels of the nervous system. We have our spinal cord, which connects the PNS to brain and reflexes. We have our lower brain or subcortical level, which is for subconscious activities. Okay, so this is for control of breathing, heart regulation, wakefulness, and we have the cortical level, which is for the memory storehouse. So everything higher level processes, thorough processes, awareness, consciousness. And our cerebral cortex actually has 47 functional areas called Brodmann areas, which we will be talking about further on as well. And we have primary. So primary is they have direct connections with muscles or directly detect specific sensations, and we have the secondary areas which interpret or process the information from the primary areas. Let's now talk about the layers of the cerebral cortex. So in general, we have 6 layers. We have first the molecular layer or the plexiform layer. Next, we have the external granular layer. Okay, fine, and then we have our external pyramidal layer, and then we would have our internal granular, and then our internal pyramidal layer or our ganglionic layer, and then our multiform layer, which is a layer of polymorphic cells. Alright, so I've summarized everything in a table form for the layers of the cerebral cortex so that we would have an easier time understanding them. We're going to start first with the molecular layer, or our first layer. As you can see right here, this would contain non-specific afferent fibers from the cortex or the thalamus, and they have horizontal cells of Cajal. Alright, so if we are going to look at them microscopically, this is how they're going to look like right here. Okay, so afferents, which means where they're coming from, is from the cortex and the brainstem, which will bring them to the other regions of the cortex because they have that intracortical association function. Okay, moving forward, we have our external granular layer. Okay, so this is a dense layer of stellate cells that actually connects one neuron to another. So these have dense stellate cells. So stellate, they look like stars, and we have small pyramidal cells in between. So they come from, again, other cortical areas in the brainstem going to other regions of the cortex. Next, we have the third layer, which is our external pyramidal, which is composed of loose stellate cells and medium-sized pyramidal cells. So they come from the brainstem, again going to the other regions of the cortex. And then we have our internal granular layer, which is composed of dense stellate cells. So our internal granular layer is closely packed and they are horizontally arranged. So they receive thalamocortical fibers from the thalamic nuclei of the ventral tier. So we have the VPM and the VPL. So these terms we will be familiarized with them almost at the end of our lecture when we start to talk about the thalamus and the hypothalamus. So they would receive input from the lateral geniculate body in the striate cortex from area 17, and it's again going to bring it to the other regions of the cortex. All right. And then we have our fifth layer. We have our internal pyramidal or our ganglionic layer. This has fewer but larger pyramidal layer or pyramidal cells. So this gives rise to the cortical bulbar, corticospinal, and corticostriatal fibers. And they actually also contain giant cells of Betz, which are projection fibers of the corticospinal or the pyramidal tract. So they again would come from the other cortical areas from the brainstem. Now this time they would go to subcortical structures. And finally we have our multiform layer, which has fusiform cells and modified pyramidal cells, and they would receive from the brainstem and they would bring it to the thalamus. So they have irregular fusiform cells, and they are actually the deepest layer of the cortex, and they give rise to projection, commissural, and association fibers. So these layers, this multiform layer is the major source of corticothalamic fibers. So again, in general, we would have first the molecular layer, our external granular layer, our external pyramidal layer, internal granular layer, our internal pyramidal layer or ganglionic layer, and our multiform layer. Okay, we're now going to talk about the lobes of the cerebral hemisphere. We're sort of done with the basics of the brain or the cerebrum. And a little tip before we dive into this is that it's really just going to be a lot about memorization, or not even memorization. I suggest that you try to just familiarize the parts of the brain, and it might seem that it's repetitive and it becomes a bit overwhelming. So try to pause, try to take breaks, because we're really going to go through it a lot. We're going to see the same areas over and over again, and the more that we see them, the more that we familiarize with them, and the more that the easier it gets for you to understand how the brain works, how everything is organized. So when you're ready, just take a pause first, and when you're ready, we're gonna dive into the topic. All right, so this is our frontal lobe. So if this is the posterior, this is the anterior. So everything here, that is your frontal lobe. Okay, so our frontal lobe is situated in the precentral gyrus and includes the anterior wall of the central sulcus and the posterior parts of the superior, middle, and inferior frontal gyri. So we have our sulci. We have our precentral sulcus, which runs parallel to the central sulcus. If this is our central sulcus, which is, if you can recall, what is the other name for our central sulcus? It's our fissure of Rolando, correct? So our central sulcus is our fissure of Rolando. Now, just parallel to that, we have our precentral sulcus right here. And in between that, you will find you guessed it right, your precentral gyrus. Okay, so this is our precentral gyrus. So it would have a superior frontal gyrus and a middle frontal gyrus and an inferior frontal gyrus. These three right here. Now these are all separated by two. We have our superior frontal sulcus and our inferior frontal sulcus. Okay. And then we will have our superior precentral sulcus and our inferior precentral sulcus right here. So that's how our frontal lobe looks like, how it's separated, the gyri that it has. And the functions of our frontal lobe would include behavior and emotion, intelligence and judgment, voluntary movement of the eyes and the body. It's also for speaking, it's for our personality, and of course for executive functions. So our executive functions are a set of cognitive processes that facilitate thinking, which is necessary for the control of behavior. So we have paying attention, organizing, planning, regulating emotions, decision making, inhibition, self-awareness, even problem solving. Okay, now let's move forward with our parietal lobe. So our parietal lobe occupies the area posterior to the central sulcus and superior to the lateral sulcus. So This is our anterior again, this is posterior, this is our central sulcus or the fissure of Rolando, and behind that would be our post-central sulcus. So the sulci of the parietal lobe, we have our post-central sulcus and our intraparietal sulcus, and behind the post-central sulcus you will find your post-central gyrus. Okay, so this is our post-central gyrus right here. There we go. And then you will find your intraparietal sulcus, and above that you will find, superior to that, you will find your superior parietal lobule or your gyri, and then your inferior parietal gyri. Okay, so if you can see the asterisk right here, so we have one asterisk here, which is, this is your supramarginal gyrus, and this one right here around this area, that is where your angular gyrus lies. So your angular gyrus is posterior to your supramarginal gyrus. So what are the functions of our parietal lobe? Well, it is for tactile and proprioceptive sensation, for sensory integration, for goal-directed voluntary movements, left and right orientation, and perceptual function. Okay, so this would mostly be about sensation. If we're talking about our post-central gyrus or our parietal lobe, it's mostly about sensation. Alright, now let's move forward with our temporal lobe. The temporal lobe occupies the area inferior to the lateral sulcus. So this highlighted area right here, that is your temporal lobe. Okay, so we have our sulci, our superior temporal sulcus, and our inferior temporal sulcus. Now, found between your superior temporal sulcus and your inferior temporal sulcus is your middle temporal gyrus. Above the superior sulcus, you have your superior temporal gyrus, and below or inferior to the inferior temporal sulcus, you will find your inferior temporal gyrus. Alright, now there's another very important gyri here, or gyrus, which is our Heschel's gyrus. We can't see it in this view, but we can see it if we slice the brain and look at it in the anterior or posterior view. If we open it up at the lateral side, okay, it's found within the lateral sulcus. You're going to find there, the one in red, that is your Heschel's gyrus. Gyrus. Now, why is this important? Because the Heschel's gyrus contains the primary auditory cortex, and this is actually, again, entirely hidden within the sylvian fissure or your lateral sulcus. Okay, so that is your Heschel's gyrus. Okay, finally we are on to our fourth lobe. This is our occipital lobe, which occupies the small area behind the parieto-occipital sulcus. Okay, so this is our parieto-occipital sulcus. This, everything right here, that is your occipital lobe. And we have our calcarine sulcus right here that separates. If you remember a while ago, we have the inferior lingual gyrus and the superior cuneus. And the main function of the occipital lobe is for your vision. Alright, now we are done talking about the lobes of the brain. And remember a while ago when we were talking about the cerebrum, we said that it has 47 functional areas called your Brodmann areas. So we have our primary Brodmann areas, our secondary Brodmann areas, and our multimodal association areas. Now, this is the part where we have to talk about that. We're going to be talking about our Brodmann areas now. So this is a huge bulk of information, so I would suggest to take a pause and breathe first, and when you're ready, we're going to head right on to it. Okay, so the way we're going to talk about the cortical areas is as divided by the lobes. So we're going to talk about first the Brodmann areas that are found within your frontal lobe, parietal lobe, temporal lobe, and so on. Okay, so our cortical areas, these are divisions of the cortex into different areas of specialization as described by Brodmann. And the main cortical areas are named after their anatomical locations, okay? we'll talk about first our primary motor area. So we're gonna start with everything in the frontal lobe. So we'll talk about 6 Brodmann areas in the frontal lobe. Okay, first is our PMA, or our primary motor area, which is area number 4 right here. All right, so this produces isolated movements on the opposite side of the body, as well as contraction of the muscle groups concerned with the movement. Isolated ipsilateral movements usually do not occur in this area once you stimulate it. However, bilateral movements of the extraocular muscles of the upper face, the tongue, the mandible, the larynx, and pharynx could occur. So again, when we talk about area 4, the primary motor area, this is responsible for firing of muscles. Okay, this is responsible for execution of movement. So speed, strength, the actual firing, the immediate activation of the body. So that is what your area 4 is for. And this is responsible for your corticospinal tract. So what happens if there is a lesion in area 4? So what could happen? If there are impairments, paralysis, spasticity, paresis, and weakness could happen in the body. Next, we have number 2, our area 6, the supplementary motor area, or premotor cortex. So these are secondary association areas. So we'll first talk about our supplementary motor area, which is found medially. So this is closer to the midline. Okay, medially, and then we have your premotor cortex, which is lateral. All right, okay, your SMA, or your supplementary motor area, focuses more on the sequencing of an intended action. So the sequence of an action. Our SMA is more responsive to visual and auditory stimulus that are relevant to the intended action. All right, so our SMA, again, it's for sequencing. So this regulates the somatosensory input into the motor cortex. Now, this does not include the execution of the action. So no execution for this one. All right, it's all about sequencing and planning. And then we have our premotor cortex, which is found laterally. So this works during patterned movements, and this initiates with our primary motor area. So this works closely with our PMA in area 4. Okay, so this plays a role in the control of axial and proximal muscles. All right, so this prepares the motor cortex in advance for the execution of the movement. And this also, sometimes we can say it's for muscle memory. Okay, now what happens when there are lesions in this area? So what can happen if there's a lesion in the premotor cortex? Nagkakaroon tayo ng apraxia. So apraxia, there is no coordination. The patient is unable to do patterned movement, but they can do it in parts kasi nawala dun yung control ng muscles natin, nawala yung responsible for patterned movements natin. Now, what happens if there is a lesion in our SMA? So, nagkakaroon ng akinesia, or walang movement. Okay, now, on our 3rd Brodmann area in the frontal lobe, we have our area 8. Okay, which is our frontal eye field. Our frontal eye field is in charge of the voluntary movement of the eye, but not as a response to a stimulus, meaning you just move your eye. So this is for contralateral saccadic eye movement. So these movements are fast conjugate eye movements under voluntary control. So when we say saccadic or saccadic movement, you have your eyes right here, you would turn your gaze to the right, so it would look, the eyes would look like this. okay, moving it that way, and then you can also move the eyes on the opposite side. So that's what we say when we're following the gaze. Okay, kind of looks like a face. Anyway, so what happens then if you impair your area 8? So of course, mawawalan lang yung kung saan siya responsible for. So mawawala yung conjugate eye movements and magkakaroon ng eye deviation kasi nawawala ng control sa isang side. Alright? So we will have later on what we call area 17, and I don't want you to confuse yourselves with that because that is for movement of the eye in response to a stimulus. Okay? Next we have area 9, 10, 11, 12. So this is the fourth Brodmann area in our frontal lobe. Again, area 9, 10, 11, 12, which is your prefrontal cortex. Now, your prefrontal cortex is a heteromodal association area. So this is in charge for your behavior, motivation, your attention, and it integrates everything, all the areas to produce that output. Now, this is important. Why? Because in anything that you do, motivation is important. Motivation is part of the movement process, okay? So when there are lesions in 9, 10, 11, and 12, what could happen is you will have your behavioral affectations. So a client could become messy, a client could become misbehaved. All right? Then we have our fifth, our area 44 and 45. This is very important. Area 44 and 45, that is for our Broca's area. This coordinates and sequences muscles to produce words or to speak. So this would involve the execution of the action, which means the execution of speaking itself. So this is connected to Wernicke's speech area via the arcuate fasciculus, okay? Later on, we're going to be talking about Wernicke's area in our temporal lobe. So if we impair area 44 and 45, what happens then? You will get aphasia or motor expressive non-fluent aphasia. So the speaking of the person will become affected. There's a difference between Wernicke's and Broca's aphasia. So for Broca's aphasia, I need you to remember that it is non-fluent, it is expressive. Okay, it is expressive, but it's non-fluent. And then we have our area 46. So area 46, this is literally just right above area 45, right here. So this is for motivation, for drive, and attention. That's it. Alright, that was it for our frontal lobe. Now we're going to be talking about the Brodmann areas in our parietal lobe. So for our parietal lobe, we have 5. Alright, so that's one less Brodmann area compared to our frontal lobe. So we're going to start with area 3, 1, 2, the primary somesthetic area. Now remember what I said about our parietal lobe, that when we talk about the parietal lobe, it's all about sensations. Alright, so our area 312 is our primary somesthetic area. This is responsible for cortical sensations. When we say cortical sensations, these are your light touch, your pain, your temperature, and it's also responsible for sensory perception. We have stereognosis, we have 2-point discrimination, localization, proprioception, and kinesthesia. Okay, so this is found in your post-central gyrus, just a reminder, and this integrates sensations felt by the body. So then it would receive sensory information from the skin, the mucosa, and other tissues of the body and the face, and it will also receive fibers that convey touch and proprioceptive input. Okay? So you will get proprioceptive input from your muscles, your joints, and your tendons. That is the function of your area 312. Okay, so if we impair area 312 or our primary somesthetic area, what could happen there is our cortical sensations may remain intact, but our proprioception and localization could be impaired. So for example, you stub your pinky toe. you can feel the pain, but you don't know where it is, or you can't localize that the pain is coming from your pinky toe. You just feel the pain, but you don't know where it's coming from. Alright, now let's talk about our area 5-7, which is our secondary sensory area. Our area 5-7 is our sensory association area, and this would receive input from areas 3-1-2, and area 7 would receive visual input from area 19. Okay, so our secondary sensory area associates feelings in its deeper meaning. So this is actually the idea of feeling and the memory part of sensation. So it's all about the memory part of sensation. Okay, so what do I mean about this? Like, for example, I hold a cylinder or a cylindrical container, and I'll be like, oh, this is my water bottle. Okay, so if we impair area 5-7, if we impair this area right here, we would have a stereognosis. So a stereognosis is that you're not unable, you're unable to recognize what it is that you're feeling. Okay, so you would get contralateral loss of tactile discrimination. You would have a stereognosis or loss of stereognosis, which is the ability to recognize form. Loss of statognosis, which is the ability to recognize the positions of your body parts in space. You can also have contralateral neglect, or there is a side of your body which you will neglect, or you won't be aware of. Okay, so you can't perceive cortical sensations, you don't know the shape of your body, you don't realize your joint positions. It's like, I know that I am touching something, but I don't know what it is. Alright, third we have our area 39. Our area 39, or our angular gyrus, which receives input from areas 18 and 19. So the angular gyrus is part of your temporoparietal heteromodal association area, and this functions closely with area 22 and 40, which is your supramarginal gyrus. So this area would integrate all sensory modalities to understand written and spoken language. Okay, so, and then we have our area 40, or our supramarginal gyrus. So this is area 40, which works closely with area 39. Alright, so this is 40, this is 39 right here, magkadikit lang sila. So our supramarginal gyrus interrelates somatosensory, auditory, and visual input. So this is for phonological processing and for emotional response. Alright, so this interrelates again somatosensory, auditory, and visual input. So let's talk about what happens if we have impairments on our angular gyrus. Now, if we impair area 39, if we impair that part right there, what will happen is that we can get agraphia or lexical agraphia, which is what we call, which is a difficulty spelling lexical words or regular words. So lexical words are all the words here. That's a normal word, that's a normal word, area, good, bad, those are your lexical words. Now we have non-lexical words, which are words that do not have meaning. Let's say words such as and all the other made up words, those are your non-lexical words. So for area 39, they have difficulty spelling normal or regular words, and they may have mild anomia. Okay, but if we impair either of area 39 or area 40, which is your supramarginal gyrus, you could get Gerstmann syndrome. So, or Gerstmann syndrome. This is composed of four symptoms. You could get agraphia. So agraphia is the inability to write and communicate through writing. You could also get alcalculia, which is sort of an inability to perform mathematical calculations. Next, we could have finger agnosia. That means there is a loss of ability to distinguish name or recognize your fingers, and you have left and right disorientation. which is the inability to distinguish left from right. So if either of area 40 or area 39 will be impaired, you could get Gerstmann syndrome. But if you impair, but if you get an impairment on area 40, you could get ideomotor apraxia, ideational or sensory apraxia, facial apraxia, and conduction aphasia. Okay, so for ideomotor apraxia, an example with this for you to understand it is, let's say I ask you to button your shirt, you wouldn't be able to do so. If I ask you to comb your hair, you won't be able to do so. So those types of if I ask you to brush your teeth, you can't do it, okay? So it's the inability to manipulate the tools with the retention of the ability to explain. So it's like when I ask you, okay, brush your teeth. You know how to brush your teeth, but you can't do it. You can say that, oh, I'm gonna get the toothbrush, I'm gonna brush it up and down my teeth, but when you try to do it, you can't. Okay, so that's classic apraxia. And then we have sensory or ideational apraxia. So what happens here, this is characterized by the inability to formulate the ideational plan. So this is the plan, okay? So this is for the plan of executing the several components of a multi-step or a complex act. For example, performing the steps of tying a shoe when asked to do so. So this occurs most frequently in diffuse cerebral degenerating disease such as Alzheimer's disease and multi-infarct dementia. And then we could have facial apraxia. So facial apraxia is an inability to perform facial oral movements on commands. When I tell you to raise your eyebrows, you won't be able to do so. And then finally, we have conduction aphasia, which is associated with poor repetition of spoken language. So when I ask you to, can you repeat dog for me? You won't be able to repeat it. All right. And then our fifth, we have area 43, our primary gustatory area. Area 43, it's in the word itself. So this is area 43. Okay, this receives input from your ventroposteromedial nucleus of your thalamus, which is for the perception of your taste. So that's area 43. If you damage this area, no area 43, you lose your taste perception. Okay, we're almost there. Now we're on to our temporal lobe. We're only going to talk about two Brodmann areas here. The first of which is your area 41-42, which is your primary auditory area. So Heschel's gyrus is here for the perception of sound, and this receives input from the medial geniculate body. Okay, so this is just an important term that we need to remember that we will be discussing when we talk about our diencephalon. All right. Okay, so when we impair area 41 and 42, what's going to happen is, of course, this is for your auditory, it's your auditory area, it's for your hearing. So if we destroy 41 and 42 on one side, it would not be detectable, but it could lead to partial deafness. But if there is an impairment on both sides of the area 41 and 42, then that could lead to cortical deafness. All right. Next, we have our second one, which is our area 41 and 42, which is what I mentioned before, our Wernicke's area. Remember, we talked about Broca's area. Now we have our Wernicke's area. So this is our auditory association cortex, and this is part of the temporoparietal heteromodal association area. The main function of this is that it discriminates frequency and sequence of sound. So Wernicke's area, this is for comprehension. Okay, for comprehension. All right, take note of that. That's for comprehension. Now, if we impair area 41 and 42, our Wernicke's area, we would get Wernicke's aphasia. So this is difficulty in understanding spoken words, but you can still understand written and environmental cues. So Wernicke's aphasia, it is sensory receptive fluent aphasia. Alright, so we're down to our last lobe, which is our occipital lobe. We have two Brodmann areas here. First, we have your area 17, your primary visual area. and partnered with area 18 and 19, your secondary visual area. So area 17 is the primary. So this is for the perception of light, the response to stimuli. Now this interprets and processes visual information received from the lateral geniculate body. Now it will then be also processed by your secondary visual area. Now for our area 18 and 19, this would extract features and would recognize shapes. So it gives meaning to what you see. That's what area 18 and 19 for. So if we alter or if we impair area 17, you could get a visual field defect or our contralateral homonymous hemianopia. And if we get an impairment in area 18 and 19, we could get depth perception impairments or dyschromatopsia. So dyschromatopsia is sort of like a color blindness. Okay, just to make it easier for everyone, again, I've summarized everything into a table form which contains the different Brodmann areas in each lobe. So this is for our frontal lobe. Again, our frontal lobe mainly for everything motor. Okay, frontal lobe, our precentral gyrus, everything motor, mostly, okay? So pause and take a screenshot. And then we have now our parietal lobe. So when we talk about our parietal lobe, everything about sensation. Okay, so again, pause, take a screenshot, and then analyze again, go back again on different Brodmann areas so you can refresh our memory. This is for our temporal lobe. Pause and take a screenshot. And this is for our occipital lobe. So you can again pause and take a screenshot. Alright, so this is our cerebral dominance. We're going to talk about this first before we proceed to the association areas of the brain. So this is almost the same as what we talked about previously about like the left hemisphere versus the right hemisphere, and then our right-sided CVA or our left-sided CVA. So the dominant hemisphere essentially is responsible for the propositional language consisting of grammar, syntax, and semantics, and this is responsible for speech and calculation. And the left hemisphere is dominant in 95% of the cases, okay? And then we have our non-dominant hemisphere, which is primarily responsible for the three-dimensional or spatial perception and nonverbal ideation. So then if we talk about the different lesions again, if we get a lesion on the dominant superior parietal lobule, you could get contralateral loss of sensory discrimination or asterognosis and contralateral neglect. Now, if we get that in our non-dominant hemisphere, we could get contralateral neglect. And then we also could get lesions on the dominant inferior parietal lobe for both hemispheres. So for the left, you could get receptive aphasia, Gerstmann syndrome, alexia with agraphia, tactile agnosia, ideomotor apraxia, and ideational apraxia. So it would be best for everyone to sort of like get a quick glimpse of how these manifest. Okay, but it's not necessary for you guys to memorize it at this point. I would just like you guys to be familiarized with the following terms, but no need to actually memorize, okay? And then for the right or the non-dominant hemisphere, we could get lesions on the inferior parietal lobe. You would get again left-sided hemi-neglect, topographic memory loss, anosognosia, constructional apraxia or dressing apraxia, and lesions on the frontal gyrus in the non-dominant hemisphere. You could get like problems with the Broca's area. So you would have expressive dysprosody or the inability to articulate the pitch and rhythm of speech. And we have also the superior temporal gyrus. So this corresponds to the Wernicke's area, which then you could get receptive dysprosody, which is the inability to perceive pitch and rhythm of speech. Let's now go to the association cortex. So the association cortex, these are areas that receive information from both the primary motor and sensory areas. So these areas have multiple inputs and outputs and are concerned with behavior, with discrimination and interpretation of sensory experiences. So if we look at the slide or the picture here, we kind of have like a main three and an additional one that we're going to talk about. So this is according to Snell. The main three association cortices are your anterior temporal, your posterior parietal, and your prefrontal association area. And then later on, we're going to be talking about our parieto-occipito-temporal association cortex. Let's start first with our prefrontal association area. So our prefrontal association area, this association area is the association of experiences that are necessary for production of abstract ideas, judgment, emotional feeling, and personality. So that is for your prefrontal association area. When we're talking about ideas, when we're talking about judgment, feelings, personality, that's everything in the prefrontal association area. Now for your anterior temporal, this plays a role in the storage of previous sensory experiences. So this is your storage. So your anterior temporal is your storage. So if we stimulate this area, this would cause a recollection of objects seen or music heard in the past. So when we stimulate this, when we stimulate the storage, you will remember memory, and that is your anterior temporal association cortex. And then we have your posterior parietal, which is for your stereognosis. So what happens in stereognosis is there is visual information from the posterior occipital cortex and sensory input of touch and pressure and proprioception from the anterior parietal cortex. Now this is then integrated into concepts of size, form, and texture, and that is your stereognosis. Okay. And then we also have body image and spatial recognition. So the brain knows where each part of the body is located in relation to its environment. And then we have our parieto-occipito-temporal association area. So this is responsible for several functions. Included are the analysis of spatial coordinates of the body, area for language comprehension. It's also for the initial processing of visual image, and this is also for the area of naming objects. Okay, so that's your parieto-occipito-temporal association area. So again, for spatial coordinates of the body, it would receive visual sensory information from the posterior occipital cortex and somatosensory information from the anterior parietal cortex. So when we're talking about spatial coordinates, it's essentially a combination of our visual, and then you would have your auditory, and on top of that would be your body surroundings. Okay, just write over this. Again, it's also for language comprehension and for initial processing of visual language or reading. So the angular gyrus again is involved in this, and this is needed to make meaning out of the visually perceived words. Now let's recall what area is our angular gyrus, if we can remember. So our angular gyrus, this is area 39, found in our parietal lobe. Okay, remember that our angular gyrus is integrating all sensory modalities to understand written and spoken language. That's why this is connected to this association cortex. And it's also the parieto-occipito-temporal association area is also for naming objects. So names heard through auditory input and physical characteristics through visual input. Okay, let's now talk about the somatotopic organization and our homunculus. So the homunculus is a topographical organized map of the proportional representation of the contralateral somatosensory or motor neurons on the cortex. Okay, so we have our motor homunculus and our sensory homunculus. Okay, now Or just listen carefully here. I'm just going to explain how everything works. So let's start with our motor homunculus. So starting from below, if we look at below, passing superiorly, so superiorly going up, we have here involved the structures for swallowing, okay? Going up, and the tongue, the jaw, the lips, the larynx, the eyelid, and the brows. And the next area, so we're already here, the next area is an extensive region for the movements of the fingers, okay? We have movements for the fingers, especially the thumb, the hands, the wrist, and then you have your elbows, your shoulders, and your trunk. Okay, so the movements of the hip, knee, and ankle are represented in the highest areas of the precentral gyrus, and the movements of the toes are situated on the medial surface of the cerebral hemisphere in the paracentral lobule. So the movements of the anal and the vesical sphincters are also located in the paracentral lobule, and the area of cortex controlling a particular movement is proportional to the skill involved in performing the movement, and this is unrelated to the mass of the muscle participating in the movement. So it doesn't mean that the bigger the muscle, that the more that the brain function is needed. So it's all about the skill involved in performing the movement. So what are we saying about the skill? So the larger it is, again, this is a topographical organized map. So the more skill needed to move the body part, the bigger its representation in this homunculus. Okay, so let's look at our sensory homunculus now, starting naman from the medial. Okay, we're gonna start from the medial now. All right, so starting from the medial, again, medial is malapit sa midline. So we have the postcentral gyrus. The area corresponds to the sensory territory of the genitals. So going up here, this is your genitals, going lateral. Okay, so moving more laterally along the postcentral gyrus, superior to the superolateral regions of the gyrus would correspond to your neck, to your shoulder, and your torso, while along the lateral side, the region corresponds to the upper extremities and then the hand. And then the inferolateral region of the postcentral gyrus would correspond to the facial and our bulbar structures. So interestingly, the face is the structure that takes up the most area of the sensory homunculus. So if we look at this illustration, that's going to make sense now why the lips and the tongue are so large and the knee is so small, because your lips and your tongue, they are specialized for making small, precisely controlled movements like when you speak, whereas your knee makes larger, less precise movements. Okay, so that's how it is. And we have more, there's a lot of sensation when we taste something. There's a lot of sensation in our hands compared to our feet. Okay, so this one takes up a bigger area compared to your feet, a very small area for your knee. So that means we use more sensation on our hands, on our tongue, in our face. All right, so that's our topographical organized map, our homunculus. We're nearing the end of our telencephalon. We're down to the subcortical white matter. Now, our white matter, these are axon fibers and they transmit impulses to the spinal cord. And our gray matter, these are cell bodies to processes. Okay, so in our subcortical white matter, we're going to be talking about 3 types of fibers. First, we have our projection fibers and our commissural fibers, and then our association fibers. So our projection fibers, they are from the higher centers down to the spinal cord. Our commissural fibers are fibers from one hemisphere to another hemisphere. And our association fibers are connections within the same hemisphere. Okay, so projection fibers, if this is, let's say that's our brain, okay, just think about it, that's your brain. These are from the higher centers down to your spinal cord. Okay, our commissural fibers are from one hemisphere to another, and our association fibers are connections within the same hemisphere. Okay, within the same hemisphere. Okay, let's start our projection fibers. We have our corticofugal or fugal, and then our corticopedal. Okay, we're going to focus on our corticofugal. We have our internal capsule, which is the upper part of the brainstem. We have our corona radiata, which is the radiation in the cerebral cortex, and then our optic radiation. So there's nothing much to memorize here or nothing much to familiarize with. We're just going to look at it right here. So these are your projection fibers. So I just need you guys to understand where they're coming from and where they're going so we can understand the functions. Okay? And then we have our commissural fibers. So we've talked about this before, our corpus callosum, which is our largest commissure. Okay, this is our corpus callosum right here. So again, when we are talking about commissural fibers, this is from one hemisphere to another, right? So this is the largest, and when we have a lesion on the corpus callosum, so mapuputol yung connection between the left and right hemisphere, it could result to an alien hand syndrome, or you suddenly your brain doesn't have control with your hands. Okay, that could happen. Okay, and then within your commissural fiber, again, we have our anterior commissure. For our anterior commissure, this connects the olfactory cortex and the temporal lobes, and then we have our hippocampal commissure, and this would then connect the hippocampus to the either hemispheres. Okay, and then finally we have our association fibers. So association fibers, again, these are fibers that run within the same hemisphere. It's quite a lot. So we have 1, 2, 3, 4, 5, 6, and 7 fibers. So I just need you guys to understand where this connects to where. So this is the main point of this part of the topic. So our subcortical white matter. So our uncinate fasciculus, this connects the frontal lobe with the temporal lobe. Okay. Next, our cingulum, this connects the frontal and parietal lobes with the parahippocampal and adjacent temporal cortical regions. Next, we have our arcuate fasciculus that connects the frontal and temporal lobes. Okay, almost the same with your uncinate fasciculus. Now, this is important for a main emphasis, give emphasis here. This is important for speech production. So, if we get a lesion here, you could get conduction aphasia. Alright? And then we have our superior longitudinal fasciculus. This connects the anterior part of the frontal lobe to the occipital and temporal lobes. We have naman our inferior longitudinal fasciculus, which connects the temporal and occipital lobes, and our occipitofrontal fasciculus that connects the frontal lobe to the occipital and temporal lobes, and finally your U fibers that would connect your adjacent gyri. Okay, so it's quite a lot of connections kasi this is our association fibers. This runs within the same hemisphere. Now, this is where we can understand the significance of all these parts and fibers of the brain. So because of that, the brain is such a magnificent part of our human body, and it's able to do so many powerful things because of all of these parts, of all of these processes, of all of these areas. Okay, so that is the end of the telencephalon part, and we're now going to move to our diencephalon. Alright, here we are on our last part. We're going to be talking about the diencephalon. Okay, so again, much like our cerebrum, it's very hard to find ways to not to memorize or not to familiarize, at least. So it's gonna be, again, disclaimer, a lot of information. So information overload. So the only thing we can deal with this is we take pauses, we review and review, we go back to the topics, we go back to the concept again and again. Just repeat again and again. Okay, it can help to see the whole picture first and then go back to the specifics. Okay, so let's start. Our diencephalon, this consists of the third ventricle and the structures that form its boundaries. The midline structure, with right and left halves, is separated by the third ventricle. Okay, we have our hypothalamic sulcus, so it's present there. That separates the thalamus and epithalamus, and also the hypothalamus and subthalamus. So it's also like the cerebrum or the cerebral cortex divided into two halves. So again, your hypothalamic sulcus is a small groove on the lateral wall of the third ventricle that separates the thalamus and epithalamus, the hypothalamus and subthalamus. Now, let's proceed to the parts and functions. No, just something very basic with the parts and functions. So, the thalamus is the relay center. Remember that, the relay center for sensory inputs to the cortex. Next, we have our hypothalamus. Now, our hypothalamus, if you were able to grasp in the previous slides, so this is for the regulation of our homeostasis. Next, we have our pineal gland in our epithalamus, which is for our sleep-wake cycle or circadian rhythm. And then finally, we have our subthalamus, which influences motor output. So just try to remember those four general functions of the thalamus, hypothalamus, the pineal gland, and the subthalamus. Okay, so this is our subthalamus. So we have our subthalamus right here, and it has the red nuclei and the substantia nigra for movement regulation, along with the rest of the basal ganglia. If you remember, you just previously said that our subthalamus influences motor output. Now, it does that by working together with the basal ganglia, and it would have your red nuclei and the substantia nigra, okay? Next, we have our epithalamus, okay? Our epithalamus consists of the habenular nuclei and their connections to the pineal gland, all right? So, the habenular nuclei is the center for integration of olfactory, visceral, and somatic afferent pathways. If you can look here, it's connected with your pineal body. Now, your pineal gland inhibits production of hormones or indirectly inhibits the secretion of releasing factors by the hypothalamus. Now, we have your pinealocytes, okay, and glial cells for the pineal gland, and the pineal secretions produced by the pinealocytes will reach the target organs via the bloodstream or through the CSF. If you recall a while ago, it's also regulates melatonin release, okay, which is important for our circadian rhythm. Alright, now we're on to the bloody part of the topic. This is our thalamus. Our thalamus is found deep within the brain. It's a large ovoid mass of gray matter that forms the major part of the diencephalon, and it serves as a cell station to all main sensory systems except the olfactory pathway. So our thalamus essentially integrates visceral and somatic functions of the body. Now, if you look at the picture below, this picture right here, you would see that there are several subdivisions of our thalamus. So let's start with your anterior thalamic nuclei, which is this one. All right. Next, we have your dorsomedial nucleus, okay, or your mediodorsal nucleus. Next, we have your lateral thalamic nuclei, which is this one takes quite much of the space of the thalamus, okay? So your lateral thalamic nuclei is further subdivided into a dorsal tier and a ventral tier. So our dorsal tier we have three: our lateral dorsal nucleus and our lateral posterior nucleus and your pulvinar, which is not seen here. And then we have our ventral. So our ventral has our ventral anterior nucleus, our ventral lateral nucleus, and our ventral posterior nucleus is further subdivided into ventroposteromedial and ventroposterolateral nucleus. All right, and also note this: our lateral geniculate body and our medial geniculate body, which we have a little bit, talked about in the past slides. Okay, now let's sort of like briefly talk about the general functions of each of the nucleus of the thalamus. We're going to start off with the anterior thalamic nuclei. So our anterior thalamic nuclei will receive connections from the cingulate gyrus, okay? Now, our cingulate gyrus is interconnected with our limbic system. Okay, it's deeply connected with our limbic system. Now, the cingulate gyrus is found again in the limbic system, and it's involved in processing emotions and behavior regulation. And it also helps to regulate autonomic motor functions. Okay, so it works closely with the limbic system and is concerned with emotional tone and the mechanisms of recent memory. So that is your anterior thalamic nuclei. So when we think of the anterior thalamic nuclei, we think about limbic system and recent memory and emotion. Okay, next we have our dorsomedial nucleus. Now our dorsomedial nucleus, or our mediodorsal nucleus as seen in this picture, it receives connections from the prefrontal cortex. Now this one receives from the prefrontal cortex and the hypothalamic nuclei. So it is interconnected with the other groups of the thalamic nuclei, and this is responsible for the integration of a variety of sensory information that would include your somatic, your visceral, your olfactory information, and the relation of this information to one's emotional feelings and subjective states. Okay, so prefrontal cortex, dorsomedial nucleus, responsible for integration of sensory information. Okay, mediodorsal, sensory. Alright. Okay, moving forward, we're now going to talk about our lateral thalamic nuclei. Let's start with the dorsal tier. We have our lateral dorsal nucleus, which is actually a multimodal nuclei. Okay, when we say multimodal, it means that you get information from different sensory modalities. So they have projections in different areas of the brain, specifically the parietal area, where there is integration of sensation. All right. Next, we have our lateral posterior nucleus. And as I said a while ago, you weren't able to find the pulvinar because the lateral posterior thalamic nuclei is also termed your pulvinar. Okay. Now, your LP, or your pulvinar, is very important because it helps with determining visual saliency. Okay, so this one, vision. Okay, visual saliency and visually guided behaviors. All right, now this works alongside the dorsal portions of the posterior thalamic nuclei. And now we have our ventral group, our ventral tier. Let's start with our ventral anterior nucleus, which is a motor relay nuclei. So deeply related with area 6 and 8. So anything to do with movement and motion. It relays motor information from the thalamus and input from cerebellum and to the globus pallidus to area 6. Okay, so this one, ventral motor. Okay, now our ventral lateral nuclei, deeply related to area 4. Now, it influences motor activity of the cerebral motor cortex. And finally, we have our VPM and our VPL. Okay, our ventral posteromedial and our ventral posterolateral. So, our VPM, this one, okay, let's write it here. Our VPM, deeply connected with area 312. So, this is for pain perception on the face. Okay, when we say VPM, pain perception on the face. So this relays common sensations to consciousness. And then our VPL, this one naman, again, so if the VPM is to the lateral 312, VPL is the median 312, and this is pain perception on upper and lower extremities and the trunk. Okay, VPM face, VPL UE LE trunk. Okay, moving forward, our thalamus has other nuclei. We have one, our intralaminar nuclei, our midline nuclei, our reticular nucleus, our medial geniculate body, and our lateral geniculate body. Now, our intralaminar nuclei influences the levels of consciousness and alertness. For our midline nuclei, well, for this one, the functions are unknown. And then our reticular nucleus, this may be concerned with a mechanism by which the cerebral cortex regulates the thalamic activity. Then we have our medial geniculate body, which receives auditory information for both ears, but mostly on the opposite ear. And then we have our lateral geniculate body, which receives visual information from the opposite field of vision. Now, if you look at the picture here to the right, you may be confused. Why is there so many arrows here? Like, what's going on here? Well, essentially, this picture right here shows the general process on how our thalamus works. So, if you remember this part right here, this is our ventral lateral nucleus, right? Okay, so that area of the nucleus, our ventral lateral nuclei, would get information from these. Okay, and then they would integrate that and then carry it out to area 4. Okay, so that's how your thalamus works. Okay, so just a summary again, I put everything in a table so that it's easier for you guys to understand and to remember. Please reread again and go through all the information again so you are able to familiarize with the concepts. Okay, so this is our anterior group with the anterior thalamic nuclei. This is our medial nuclear group with our dorsomedial nuclei. Our lateral group for the dorsal tier, dorsolateral nuclei. This is our lateral group, the ventral tier, with the ventral anterior nuclei, ventral lateral nuclei, ventral posteromedial, and ventral posterolateral. And then we have our midline nuclear group with your centromedian nucleus and your interthalamic adhesion, and then your posterior nuclear group with your pulvinar, your medial geniculate body, and your lateral geniculate body. Okay, so another way to sort of familiarize yourself with the thalamus is based on its functional division. So we would have our sensory, our motor, our limbic, our multimodal, and our intralaminar. So we have for our sensory, these are for specific or finer sensations of pain. So what the areas that will work here, or the nucleus that will work here, would be your lateral geniculate, your medial geniculate, your VPL, and your VPM. For motor, they would convey motor signal from the cerebellum and the globus pallidus to the precentral motor cortex for finer motor planning. So what would work here will be your ventral anterior and your ventral lateral, which again, if you recall, this one is for area 6 and 8. This one is for area 4, right? And then we have our limbic, which is associated with emotions. And we have an asterisk right here. This is our anterior limbic nuclei, which is interposed between the mammillary nuclei. And this is responsible for the recollective or episodic memory. Let's write that down here. Episodic memory. Okay. And then we have your multimodal, your pulvinar, posterolateral, and dorsolateral. And finally, your intralaminar with your reticular, your centrum medianum, and your intralaminar. So for the clinical correlations of our thalamus, so what happens if there is a lesion in our thalamus or something happened to our thalamus? Well, we could have a thalamic syndrome, which is where disruptions of one of the pathways of the brain that affects the sensation of temperature. Now, when you get thalamic syndrome, you could get hyperpathia, athetosis, posturing of the hand, immediate threshold of sensitivity to pinprick, heat, and cold. So it's mainly with like affectations in our temperature. Okay, so this is different from hyperalgesia. So your hyperpathia, because hyperalgesia is painful sensation with a noxious stimuli. Hyperpathia is just everything is just painful. Even if it's non-noxious, it's painful for you. Okay. All right, we're on to our last topic, which is our hypothalamus. Now, this is all based on Snell, so please refer to Snell. Our hypothalamic nuclei, or our hypothalamus, nearly all the activities in the body are influenced by the hypothalamus. So our hypothalamus controls and integrates the functions of the ANS and the endocrine systems and play a vital role in maintaining homeostasis. So the main functions, it regulates your body temperature, your body fluids, your drive to eat and drink, your sexual behavior, and your emotion. Now, our thalamus is divided into two. It's divided by an imaginary parasagittal plane into medial and lateral zones. So we have a medial zone and we have a lateral zone. Now, it's quite extensive, and again, the subdivisions or the divisions of the hypothalamus isn't really defined. So you would find, let's say, part of the preoptic nucleus. You'll find that in the medial, but then you'll also find it in your lateral. So these are your hypothalamic nuclei. So if we look at the picture to the right, we have again part of your preoptic nucleus, and then you would have your anterior nucleus, this one right here, and then a part of your suprachiasmatic nucleus, your paraventricular nucleus, and then you would have your dorsomedial nucleus, kadikit lang sila, your ventromedial nucleus. You also have your arcuate nucleus and your posterior nucleus. Okay, this is different. This is a posterior pituitary. This is your posterior nucleus right there. Okay, so that's for the medial part. And then we have your lateral part. So again, it's going to cover the preoptic nucleus again, and then a part of the suprachiasmatic nucleus again. You will find your supraoptic nucleus, your lateral nucleus. So that is your lateral. This one is lateral preoptic. Your lateral nucleus is unfortunately not seen in this picture right here. Your tuberomammillary nucleus and your lateral tuberal nuclei, which is also not found in this picture. Our hypothalamus is actually very extensive. So what we're going to do now is, again, we've just summarized it in a table right here where you can just find the zone that it comes from, the name of the nucleus, and its presumed function. So again, you can just pause and just screenshot, and then you can have it to review the functions over and over again. Okay, when you're done taking a screenshot, we're going to proceed to a quick okay, when you're done taking a screenshot, we're just going to briefly discuss all of them. Okay, so we're going to start with your medial and your lateral. So your medial and lateral would have your preoptic nuclei and your suprachiasmatic nuclei. Again, they're together because part of them would be part of the either zone. So your preoptic would control parasympathetic system. Your suprachiasmatic controls your circadian rhythm. Okay, and then we have your medial. Your medial has your anterior nuclei, your paraventricular nucleus, your ventromedial nucleus, your medial hypothalamic nuclei, your arcuate nucleus, and your posterior hypothalamic nuclei. Your anterior nuclei helps with temperature. So I need you to remember anterior nuclei is for temperature. So this is response to heat, and this is your heat loss center. Your paraventricular nucleus synthesizes oxytocin, ADH, and ACTH. Your ventromedial nucleus is for the regulation of emotion, or for passivity, so being passive. Your medial hypothalamic nuclei will be your satiety center. So this will inhibit you from eating and reduce food intake. So your medial hypothalamic nuclei will tell you, hey, stop eating, you're full. However, your lateral hypothalamic nuclei will tell you to, hey, you're hungry, go eat. So this is your eating center, your hunger center, and it's also your thirst center. Okay, so your medial is like, hey, stop eating, you're full. Your lateral hypothalamic nuclei says, yo, you're hungry, go eat. Okay, and then we have our arcuate nucleus, which is for the regulation of action of the hypothalamus, and this also helps with the production of dopamine, and it stops prolactin production. Your posterior hypothalamic nuclei is also for temperature. Okay, now this one regulates temperature, that is response to cold. If your anterior is to heat, your posterior is to cold. Okay, and then your supraoptic nucleus, which synthesizes vasopressin, which is your antidiuretic hormone right here. Right. Now, what happens if we get a lesion from your hypothalamus? Well, generally, the most common clinical disorders with hypothalamic lesions, it's either you get severe obesity or wasting, you get genital hypoplasia, you get hyperthermia or hypothermia, diabetes insipidus, disturbances of sleep, or emotional disorders. Okay, so what could happen if there is a lesion in your paraventricular nuclei? So again, your paraventricular nucleus synthesizes oxytocin, ADH, and ACTH. So if there is insufficient production of ADH, your antidiuretic hormone, what will happen is you will have increased frequency of urination. Okay, and then you keep on urinating, you get dehydrated. Eventually, you could get diabetes insipidus. Now, what would happen if you get a lesion to your anterior nuclei? So again, your anterior nuclei, anterior temperature, anterior heat. This is your heat loss center. Anterior nuclei is for your heat loss center. So you would have the inability to lose excess heat. You get hyperthermia. Now, what would happen if you get a lesion to your ventromedial nuclei? So your ventromedial nuclei is for regulation of emotion, your passivity. So if you destroy that, your ventromedial nuclei, you could get rage. Okay? And then your ventromedial nuclei is also for food intake, right? So it's also for your satiety center. Now, if nasira yung satiety center mo, okay, you would get hyperphagia or chronic excessive hunger, and that can lead to obesity. Okay, so see how all of them are sort of like in the same general category as those. Now for your lateral nuclei, so your lateral nuclei is for again your hunger center. So if masira yung hunger center natin, what could happen? It's again just the opposite of what happened a while ago. So if we destroy the lateral, you would be opposite of obese would be anorexic. Okay? You have lack of appetite. Down yung appetite mo. And also, you would have lack of desire to drink water. What happens now if we get a lesion to our suprachiasmatic nuclei? So our suprachiasmatic nuclei is for your circadian rhythm, your sleep-wake cycle. Okay, when that is broken, you could either get insomnia or, what's the other one? Too much sleep would be hypersomnia. Okay, there we go. So that is the end of our topic. We're done with everything else. We're done with the telencephalon, the diencephalon, and that's the end. If there is anything that you didn't understand, I hope that you were able to take note, to write them down. This topic is really a lot of concepts, a lot of memorization or familiarization of terms, of where the brain, this part of the brain is located, what does this thing do. But again, it's just through a lot of repetition that you'll be able to master it faster. Okay, so if you do have questions, please do take note of them and we can discuss them together, all right? Thank you very much for your attention. It's been great, and I hope you learned a lot from this, and these are my references.